So guys, here we have two plants, and these are the ones I'm going to use in my presentation. So we have the Lantana Camara here. We have the Lantana Camara. Can you see? Lantana Camara. Can you see it very well? The Lantana Camara. Okay. With the yellowish leaves. Alright. So we are having a twig of that plant. Then here we have the Trandescantia, that one with the purple, that is the Trandescantia, and these are the plants we are going to look at in the practical. In one of the questions for this practical, we'll be having these two plants as specimens. The first plant is that one, a twig of that plant there. And that plant, this plant is known as the Lantana Camara. So you can see there's thorns, all right? There's thorns, and it has flowers, purple and yellow flowers, all right? Then the second one we are going to look at is this other plant here, known as Trandescantia, and you can see it has purple leaves. So we are dealing with these two specimens in our practical today. So there will be questions on those. So guys, I just wanted you to see them physically, the way they look like. Good morning, my audience, my students, and all the people who are watching this video. I continue with my biology practical papers, marking, and today we have an interesting practical from the KCSE 2014. The KCSE 2014 and the 3 questions, and the first question was testing the behavior of yeast cells. As you can see there, that's an yeast cell. Question number 2 was diving into the knowledge of people knowing about the plant Lantana Camara and the plant Trandescantia, as you can see them there. Then finally, question number 3 was a photograph of bones, and we will know the details more from the video as we progress with the details of the practical. So, let me start by introducing myself. My name is Teacher Mr. Midamo, and I am having a YouTube channel. And in this YouTube channel, I post videos, and some of the videos I post are the ones which are dealing with biology practical series. Why biology? Biology is because I was an examiner for a very long time, and I retired examination duties in 2016. So I've been an examiner and a senior examiner at the same time, a trainer of the examiners. So in today's video, I've reached the year 2014, and this is a very interesting practical because we are dealing with a very interesting living organism which is used in very many areas. And even in this morning, I took my breakfast and used a product of that living organism. You get to know the details of it as we progress with the practical. Otherwise, my question is this. Have you ever wondered how the simple fermentation process we see in everyday life connects to one of the most fascinating aspects of cellular biology? We will dive in and uncover the science behind the bambos. In this video, you master the concept of aerobic respiration through hands-on experiments, guided by a senior examiner, Mr. Midamo. By the end, you not only understand the scientific process, but also gain practical skills to ace your biology exam. Have you ever baked bread or brewed a drink and you noticed bambos forming? Or have you visited the Kenyan breweries or the Broadway Brand Factory? Those bambos are more than just a product of cooking. They are the result of a fascinating biological process that happens within the cells. In today's experiment, I'll guide you through how yeast turns sugar into carbon dioxide or carbon dioxide and alcohol using tools from the lab. And we will usually prove this reaction. Whether you are a student or just curious about biology, this experiment will open your eyes to the science happening in everyday life. Here, we move and start. So, here is the illustration of a yeast cell undergoing bonding, showing key parts such as the parent cell, the bud, the cell wall, and other internal structures. Remember, yeast is a living organism. In equation 1, which is now dealing with the yeast, you are provided with solutions labeled Q and R, a substance labeled D, and a delivery tube fitted with a rubber bang or cork. Roman 1, label solution Q as lime water. 2, you label solution R as 10% sugar solution. 3, add substance D to the 10% sugar solution. 4, tightly close or plug the boiling tube with a rubber bang or cork fitted with a delivery tube. 5, dip the end of the delivery tube in the test tube containing the lime water. 4, put the boiling tube in warm water bath at 40 degrees centigrade and allow the setup to stand as shown in the diagram below. So all those apparatus, you are supposed to assemble them and they will look like what I will show you below. So that is the setup. And you are supposed to observe the setup for 15 minutes. So you can see the delivery tube, the test tube, the lime water, cork, boiling tube, substance D, and the sugar solution mixture, warm water, and the beaker. That is the way you set up the experiment. State the observations made in the lime water is your first assignment. Explain the observations made in the lime water is your second assignment. Name the physiological processes that was being investigated, your second or your third. Write a word equation for the physiological process investigated. And finally, why was the warm water bath used in the experiment? So in B, so that was those were the questions. You are, after assembling the apparatus and waiting for 15 minutes, you put and drop of the contents in the boiling tube on a microscope slide. Stain with a drop of methylene blue and cover with a cover slip. Observe it under a light microscope using low, medium, and high power objective lens. Draw and label one of the structures observed under the high power objective lens. And in Roman 2, you state the magnification of your drawing, and Roman 3, you state the identity of substance D. So, in equation 2, you ask yourself this. Have you ever wondered how two simple plant specimens can reveal the secrets of plant classification? We will dive into the world of Lantana camara and Tradescantia and uncover the fascinating clues hidden in their structure. By the end of the video, you will not only be able to classify plants like a biology expert, but also identify key adaptations that allow them to thrive in their habitats. We will break down the observable features of Lantana camara, which was given as specimen E, and Tradescantia, which was given as specimen F. Examine their leaves, stems, and habitat adaptations, all under the guidance of a senior biology examiner. You will learn how to master plant classification for your exams or personal interests. So, That is E and F. E is lantana camara, which is on your left, and right is the transcantia, which is your F. Question 2 questions. You are provided with specimen labeled E and F. Name the subdivision to which the specimen belongs. Using features on the specimens, give 2 reasons for your answer in A1 and above. State the differences between the ribs of specimen E and F. Stems of specimen E and F. Using observable features on the specimen, state the adaptation of the stem of the specimen E to its habitat. Question 3. Ever wondered what makes animal ribs move so flexibly? Let's dive into the fascinating world of bones and joints to uncover the secrets behind their structure. By the end of this video, you confidently identify key bones in the ribs of different animals and understand the types of joints that allow their movement. Get ready to strengthen your anatomy knowledge. We will explore bones of rib as shown in the image, guided by a senior biology examiner. You will learn to identify crucial bones like the scapula, the humerus, the radius, the ulna, and the phalanges, and you discover the types of joints that make movement possible. So, the photograph below shows 2 bones, A and B, skeletal ribs of a certain mammal. And you can see the labels. There is A, J, H, B, L, K, and M. Questions you are asked in question 3. Which of the 2 A and B skeletons represents a forerib? State 2 features observable on the skeleton to confirm your answer in A1 and above. Name the bones labeled J, K, and M. Which bones forms the second joint with the bone labeled K? Name the type of joint formed at the part labeled H and L. Apart from the bones, state the function of any 2 other components of the joint. Those are the questions for question 3. Let's go to the expectations, or what you are expecting to have answered in question 1, where you had been told to state the observations made in the lime water. And the answer was bamboos, or you say gas formed, or effervescence. Reject formation of foam or froth. Lime water turns milky, or white, or cloudy, or a white precipitate, or white suspension is formed. You can accept the short form of precipitate, PPT. Roman 2, explain the observations made in the lime water. The gas produced is carbon dioxide, or carbon dioxide, or CO2, which reacts with lime water, or Q, because it was labeled Q, to form a white precipitate, or if you are good enough, you can say to form calcium carbonate, or you can say to solve insoluble salt, or insoluble compound, or insoluble solid. In this part, scoring was if bamboos or gas formed in A1 only. In A2, 1 mark is scored for gas carbon 4. oxide or CO2 and no mark for the reaction. All right. If rainwater turns white, etc., in A1, only in A2, 2 marks can be scored for gas CO2 and the reaction with rainwater. So take that precaution, my dear teacher, when you are marking that question. Question 1A summary. Observations in the rainwater. The rainwater water turns milky. Explanation for observations in the rainwater. The milkiness in the lime water is due to the formation of calcium carbonate. This happens because carbon dioxide gas is produced during the process in the boiling tube and then bubbles through the lime water. Carbon dioxide reacts with the lime water, which is calcium hydroxide, to form calcium carbonate, causing the lime water to turn milky. 1A, 2 and 4, or 3 and 4, sorry. Name the physiological process that was being investigated. The physiological process is respiration, or you say anaerobic respiration, fermentation, aerobic respiration, all those were alternatives which got a mark. Write a word equation for the physiological process investigated aerobic respiration. If you say aerobic respiration, you get that first equation there where you have glucose or sugar plus oxygen in presence of yeast or enzyme, or you can even say zymase on top of the arrow there. You have carbon dioxide or carbon dioxide plus water plus energy, or you can say plus ATP. If you wrote anaerobic respiration, you have glucose or sugar, yeast, enzyme, zymase there on top of the arrow, or it was added to the sugar, means it was added to the sugar, where you get now ethyl alcohol or ethanol, or you say alcohol plus carbon oxide or carbon dioxide plus energy or ATP. Roman 4 is tied to 3. If respiration is specified, reject CO2 since one equation is required. You are not supposed to use formula. You are instructed to write a one equation. The summary of those is the physiological process being investigated. The process being investigated is anaerobic respiration or fermentation in yeast. One equation for the physiological process is glucose plus yeast gives you ethanol plus carbon dioxide or carbon dioxide plus energy. Reason for using the warm water bath. The warm water bath is used to maintain an optimum temperature of 40 degrees centigrade for yeast fermentation. Yeast enzymes are most active at this temperature, facilitating the fermentation process. So, Roman 5, why was the water bath used? Experiment to provide optimum temperature or most suitable temperature or favorable temperature for enzyme activity or enzyme action, working or functioning or reaction or ones to that effect. Except it was put in warm water to activate enzymes. And for 5, reason for using the water bath, The water bath is used to maintain an optimum temperature of 40 degrees centigrade for yeast fermentation. Yeast enzymes are most active at this temperature, facilitating the fermentation process. Summary of question 1A. Observations in the lime water. The lime water turns milky. Explanation for the observations in the lime water. The milkiness in the lime water is due to the formation of calcium carbonate. This happens because carbon dioxide gas is produced during the process in the boiling tube and then bubbles through the lime water. Carbon dioxide reacts with the lime water, which is calcium hydroxide, to form calcium carbonate, causing the lime water to turn milky. Physiological process Roman 3 being investigated. The process being investigated is anaerobic respiration or fermentation in yeast. The equation for the physiological process, glucose plus yeast gives you ethanol plus carbon dioxide plus energy. Reason for using the warm water bath. The warm water bath is used to maintain an optimum temperature of 40 degrees centigrade for yeast fermentation. Yeast enzymes are most active at this temperature, facilitating the fermentation process. B of question 1, drawing and labeling a structure observed under the high power objective lens. So that is your drawing there. There are wrong drawings on your left there. Wrong drawing is the way you write. Wavy outline, don't allow that. Field of view of microscope, don't allow that. Wrong drawing, all right. So the drawings are there. You can have the first one there, which is cell wall, cell membrane, nucleus, cytoplasm, and a drawing mark. You can also have the alternative where you have a cell wall, cell membrane, nucleus, cytoplasm, and the drawing mark. And then you can have the third diagram, which is cell wall, cytoplasm, nucleus. All those diagrams were accepted. Rebel for that diagram with a bird, 4/2, you get 2 maximum. So you divide by 2. Drawing mark is 1, you get 3 marks. For this other one here without a bird, you have 3 rebels, and you divide by 2, you get 1.5. Approximate that 1 to 2. Drawing mark is 1, 3 marks. Rebels, no lines crossing, no arrowheads, lines touch structure, correct spelling. That's the way you get the rebel marks. Drawing mark, continuous outline, cytoplasmic inclusions, and shaded nucleus or not to get the drawing mark. Magnification, you have got there all what was expected when you used eyepiece power times 3 times 4 times 5 times 7 times 10 times 15, depending with the type of microscopes your school is having, times 40. You can see the magnifications, times 45, and the corresponding magnifications, times 50, and the corresponding magnifications, and times 65, the corresponding magnifications. And B Roman 3 is yeast or yeast cells. That is the identity of D. Question 1B summary. Drawing and labeling a structure observed under the high-power objective lens, you would likely observe yeast cells which are oval or spherical in shape, with a visible cell wall, cytoplasm, and possibly budding cells if the yeast is reproducing. Roman 2. Magnification of your drawing. The magnification would depend on the power of the objective lens used and the eyepiece lens. For instance, if using a 10 times 10 eyepiece lens and times 40 high power objective lens, the total magnification would be 400 times. Identity of substance D. Substance D is yeast, a type of fungus used in fermentation. Question 2. Name the subdivision to which the specimen belongs. Angiospermaphyta, or angiospermae, or angiospermatophyta. Accept small letters but reject wrong spelling. Using observable features on the specimens, give 2 reasons for your answer in A1 above. One is presence of flowers or fruits or seeds enclosed in fruits, obvious in flowers, and presence of veins in leaves. Summary of 2A. The subdivision to which specimen E, or Lantana camara, and F, Trandescantia, belong is angiosperma or angiospermaphyta, flowering plants. Observable features that confirm they belong to the angiosperms is that both specimens have flowers, which is a defining characteristic of angiosperms, and both of them there is presence of broad leaves and vascular tissues, xylem and phloem, observed in both specimens indicates they are vascular plants typical of angiosperms. Differentiating the leaves, B1, leaf E and leaf F, net veined, the other one is parallel veined, laugh, the other one is smooth, serrated margin, the other one is smooth margin, green, purple, thin, thick, broad, narrow, has petiole, has sheath, non-succulent, succulent, opposite, alternate. Mark correct 5 differences. Mark any correct 5. Differences must correspond. Place ticks in the middle because it is an open question. Stems, differences, stem of E and stem of F. Angular, round, rectangular, square, okay. Prickly, smooth, thorny, hairy, spiny, has hairs, spikes, has thorns, okay. Underhand, herbaceous and soft. So unde goes with herbaceous, hand goes with soft. Gray, purple, or green, purple. Leaves opposite, leaves alternate. Non-succulent, succulent or fresh or juicy. Mark any correct 2. Place ticks in the middle because it is an open question. Summary of 2B. Differences between the leaves of specimen E, Lantana camara, and F. Trandescantia. The leaves are opposite and rough textured with serrated edges. Specimen F, the leaves are smooth and have an entire margin without the teeth and are elongated. Entire means without the teeth. Differences between the stems of E and F. Lantana camara, the stem is woody and airy or thorny, providing rigidity. F, the stem is herbaceous and smooth, for example, and non-woody. Using observable features, the specimen states the adaptation of stem or specimen E to its habitat. Plique or thorny or spiny, prickly has thorns for protection against browsers or beavers or animals. Reject predator or predation or enemies. Ude or hand for mechanical support. Note, function is tied to feature, can score feature alone, but not function alone. C green for photosynthesis because it is not a xerophyte. Summary of 2C on adaptation. The wound stem provides a structural support, allowing the plant to grow upright and withstand environmental factors like wind. The preliminary thorny stem, Lantana camara, has small thorns along its stems, which help protect it from herbivores. These prickly features make it less palatable to animals, which is an important defense mechanism in its habitat. Additionally, the thorns help the plant climb and spread through surrounding vegetation. Question 3. Which of the two A and B skeletons represent a four-rib? A. State two features observable on skeletons to confirm your answer in A1 above. Tied to 1, Roman 1. Presence of scapula or shoulder blade, presence of olecranon process, presence of humerus, presence of ulna, radius, ulna, and radius. You can mention any of those. Summary of 3A. Skeleton A represents a four-rib. Two observable features on skeleton A to confirm it is a four-rib: the presence of scapula or shoulder blade, which is visible at the top right of the skeleton A. This is characteristic of shoulder region in the foreribs. The structure of the phalanges, finger bones in skeleton A. The long ridges suggest it is adapted for grasping or manipulation, which is typical of foreribs. Question 3b. Name the bones labeled J, K, and M. J is radius, K is femur, M is metatarsus. If diagram labeled indicate contradiction, or you just put CT for contradiction if the names are wrong on the diagram. 3b. The names of the bones labeled are as follows. J, humerus, the long bone in the upper arm connecting the shoulder to the elbow. K, ulna, one of the two long bones in the forearm, typically forming the elbow joint. M, phalanges, the bones that form the fingers or toes. Which bones form the second joint with the bone labeled K? Pelvic girdle, innominate bone or hip girdle or hip. Name the type of joint formed at the part labeled H and L. Grinding joint, except sliding joint. L, hinge joint. Summary of C and D. Question 3. The bone that forms the second joint with the bone labeled K is ulna. Labeled K, ulna, is the radius. It runs parallel to the ulna in the forearm and forms part of the elbow joint as well as the wrist joint. The type of joints formed at the parts labeled are H, hinge joint. This type of joint allows movement in one plane, like a door hinge, typically found at the elbow. L, ball and socket joint. This type of joint allows a wide range of motion in multiple directions, typically found at the shoulder. Apart from the bones, state the function of any two other components of the joint. component. Ligament attaches a bone to another bone. Cartilage, shock absorber or facilitate gliding or reduce friction. Synovial fluid reduces friction or nourishes lubrication or shock absorber. Synovial membrane secretes synovial fluid and crosses synovial fluid. Joint cavity contains synovial fluid. Mark any first two component is tied to function. Underline if component is wrongly spelled but mark function. Summary of 3E. The two other components of a joint and their function are ligaments. These are strong fibrous tissues that connect bones to each other at a joint. Their function is to provide stability to the joint by holding the bones together and preventing excessive movement that could lead to injury. Cartilage. This is a smooth, rubbery tissue that covers the ends of bones at a joint. Its function is to reduce friction during movement and to act as a cushion, absorbing shock and preventing bones from rubbing directly. Question 3 summary. The image displays the skeletal ribs of a mammal labeled as A and B. These appear to be four ribs, likely of two different animals. Based on the structure, they could represent homologous structure in comparative anatomy, which means they share a common evolutionary origin but may have different functions. From what is visible, A appears to have a more elongated and possibly flexible structure, potentially indicating an arm adapted for grasping or swinging, perhaps a primate or a similar mammal. B shows a sturdier, more rigid structure, possibly indicating an arm designed for supporting of movement, perhaps from an animal like a dog or horse. Each rib has been labeled with different points, H, J, K, L, M, which likely corresponds to different bones or joints. Would you like help identifying each part in detail or making a comparison between the ribs? Go and read your textbooks further.